forward with the pathway? Well, the answer to that is that the arrangement of atoms in the isocitrate is better suited for the next few oxidation reduction reactions that are about to come in the Krebs cycle. In the isomerization of citrate to isocitrate, an intermediate is also generated which is known as cis-aquanitate, but no need to go into the details of that. Moving on to the next step in the Krebs cycle. In the next step, the isocitrate is converted into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. Now we come to the first of the 4 oxidation reduction reactions in the citric acid cycle. These oxidation reduction reactions are the one which produce energy in Krebs cycle. And all of these 4 reactions use enzymes in them which are known as dehydrogenases. Now, what do these dehydrogenases do and how do they produce energy? That is very important for you to understand. See, if you look at the term itself, dehydrogenase, this literally means removal of hydrogen. So dehydrogenases are basically enzymes that remove hydrogen from a molecule. Now, as you remember that removal of hydrogen is also known as oxidation, and the hydrogen that is removed from one molecule must be passed on to some other molecule. The other molecule that receives the hydrogen gets reduced in return. So this is the basic mechanism of action of dehydrogenases. They remove hydrogen group from one molecule, oxidizing it, and in turn attaching that hydrogen group to some other molecule, leading to its reduction. This is why the reactions carried out by dehydrogenases are known as oxidation reduction reactions. So coming back to our Krebs cycle, we reached a point in Krebs cycle where isocitrate will now be acted upon by the enzyme isocitrate dehydrogenase. So let's look at what happens in this reaction. This is actually a 2-step reaction in which the enzyme isocitrate dehydrogenase first removes hydrogen from isocitrate, thus oxidizing it. The hydrogen that is removed is attached to NAD to form the NADH, thus completing the process of oxidation and reduction. But there is a small additional step that happens next, which is catalyzed by the same enzyme isocitrate dehydrogenase. In the next step of the reaction, a carbon atom is also removed from isocitrate in the form of a molecule of carbon dioxide. This leads to the formation of a 5-carbon compound known as alpha-ketoglutarate. So if you look at the complete chemical process that just happened here, this is known as the oxidative decarboxylation. Oxidative because oxidation of isocitrate takes place since a hydrogen is removed from isocitrate, and decarboxylation as a carbon group is also removed from isocitrate. The conversion of isocitrate into alpha-ketoglutarate is followed by a second oxidative decarboxylation reaction, the formation of succinyl-CoA from alpha-ketoglutarate. This reaction uses the enzyme alpha-ketoglutarate dehydrogenase that catalyzes a similar oxidative decarboxylation of alpha-ketoglutarate. In this reaction also, a hydrogen gets attached to NAD to form the NADH. Since it is also a type of decarboxylation, a carbon is removed from alpha-ketoglutarate to be lost in the form of carbon dioxide. Also, coenzyme A is used in this reaction, therefore the product will be a combination that is known as succinyl-CoA. In the next step of the Krebs cycle, the succinyl-CoA is converted to succinate. This reaction is catalyzed by the enzyme known as succinyl-CoA synthetase. Now try to understand what happens here in this reaction, as this is a very important concept and often missed by many students. The number of carbons remain the same in this reaction. Both succinyl-CoA and succinate have 4 carbons each. So what changes? Succinyl-CoA is a high energy compound, and the energy stored in this compound is used to attach an inorganic phosphate molecule to GDP. This GDP gets converted to GTP, which is like a close cousin of ATP, and is later used in the formation of an ATP. The coenzyme A attached to the succinyl-CoA also gets released in this reaction. So what happens here essentially is that the succinyl-CoA loses its high free energy and its coenzyme A to be converted into succinate and a molecule of GTP. This is an example of substrate level phosphorylation, a process of producing energy that we discussed in detail in our glycolysis video. So now we have 4 carbon compound known as succinate. In fact, the number of carbons do not change from here onwards. All the compounds which come next in the cycle only have 4 carbons. The last compound generated in the cycle is oxaloacetate, which also has 4 carbons. And that oxaloacetate combines with another molecule of acetyl-CoA to form citrate, a starting point of the Krebs cycle. Coming back to where we left the cycle. So we have succinate, which is a 4 carbon compound, and it is acted upon by the enzyme succinate dehydrogenase. As you can tell from its name, this enzyme is a dehydrogenase. So it removes hydrogen from succinate, and the hydrogen that is removed is accepted by flavine adenine dinucleotide or FAD. FAD is in turn converted to FADH, and the succinate gets converted to fumarate. FAD is the hydrogen acceptor in this reaction and not NAD, because attaching hydrogen to NAD requires more energy than this reaction can provide. So FAD is used here as it requires less energy change to get reduced to FADH. Free energy change for 4 carbon compounds is less as compared to 6 or 5 carbon compounds that were present at the first half of the cycle. There is, however, an exception to this at the end of the cycle that we will discuss in a bit. Now, the enzyme involved in the conversion of succinate to fumarate is succinate dehydrogenase, which, like aconitase, is an iron sulfur protein. This enzyme is also very important as succinate dehydrogenase is directly associated with the electron transport chain. The next step in the Krebs cycle is a hydration reaction, which means we add a molecule of water to fumarate and convert it into malate. This reaction is catalyzed by the enzyme fumarase. Nothing special in this reaction, so let's move on to the next one. The next type of reaction that happens here is the oxidation, in which we finally oxidize malate to oxaloacetate. And the enzyme involved here is again a dehydrogenase, which is known as the malate dehydrogenase. Again, the NAD molecule gets reduced to NADH, which will be used in the electron transport chain. In this reaction, NAD gets reduced to NADH. Now you may ask why NAD gets reduced here and not FAD, because like I told you earlier that the free energy change for compounds in the end of the cycle is less as compared to that was present at the first half of the cycle. Well, this was the exception that I was talking about. Even though this reaction is present at the end of this cycle, still a molecule of NAD is getting reduced here. Well, this is due to the fact that oxaloacetate that is being produced here is rapidly being utilized by combining it with acetyl-CoA to form citrate. And the second product being produced here, the NADH, is also being rapidly used in the electron transport chain. So the standard free energy for this reaction, unlike that for the other steps in the citric acid cycle, is significantly positive. I know that some students, especially in lower grades, may find some of the concepts I explain here a little advanced, but trust me, you will have to learn about the cycle in depth at one point or another, so better now. The oxaloacetate again combines with a molecule of acetyl-CoA and leads to the formation of citrate with the help of the enzyme citrate synthase. A water molecule is also added in this reaction. Now this completes one full turn of the cycle and the same sequence repeats again. Okay, so if we look at the cycle, now you can see that we got from this cycle. We added 2 carbons to this cycle in the form of a molecule of acetyl-CoA. These 2 carbons were released in the form of 2 molecules of carbon dioxide here and here. In this process of oxidation and reduction, we convert 3 molecules of NAD to NADH, 1 molecule of FAD to FADH, and we converted 1 molecule of GDP to GTP. Now there are few other important concepts regarding the Krebs cycle that you must understand to have a complete knowledge about this biochemical cycle. The first is this summary reaction of this cycle. And the second is the energetics of this cycle, meaning how many ATPs are produced for every 1 molecule of glucose that enters this cycle. So first, coming to the summary reaction of glycolysis. Now the net reaction of citric acid cycle is 1 molecule of acetyl-CoA plus 3 molecules of NAD plus 1 molecule of FAD plus 1 molecule of GTP plus 1 molecule of inorganic phosphate plus 2 molecules of water combined to give 2 molecules of carbon dioxide and 3 molecules of NADH plus 1 molecule of GTP and 1 molecule of coenzyme A. What is this gibberish? Well, let me decode this reaction for you. On the left hand side of this reaction are the raw materials that are being used here, and on the right hand side are the products that are formed in the Krebs cycle. Now in every single turn of the Krebs cycle we use 1 molecule of acetyl-CoA that combines here with the oxaloacetate to form citrate. 3 molecules of NAD are used in the Krebs cycle that get reduced to 3 molecules of NADH. Here you can see where the 3 NADH molecules are produced in the Krebs cycle. 1 FAD molecule is used which gets reduced to 1 molecule of FADH. This FADH is produced in the conversion of succinate to fumarate here. 2 molecules of carbon dioxide are produced for every 1 molecule of acetyl-CoA that enters the Krebs cycle, like I showed you earlier. 1 coenzyme A is produced in the conversion of succinyl-CoA to succinate, where succinyl-CoA loses the coenzyme A. And 1 molecule of GDP combines with 1 molecule of inorganic phosphate and leads to the formation of 1 molecule of GTP. 2 molecules of water are consumed, 1 in the synthesis of citrate and 1 here in the hydration of fumarate. Okay, so this was about the summary reaction of Krebs cycle. Now let's talk a little bit about how much energy is produced in the Krebs cycle. Most of the energy that is produced in the Krebs cycle is via the reducing equivalents, the NADH and FADH, that are subsequently utilized in the electron transport chain. If you look at the placement of these 2 biochemical pathways in the mitochondria, they lie very close to each other. the Krebs cycle operating in the matrix of the mitochondria, and the electron transport chain in the membrane of the mitochondria. So the NADH and FADH that are being produced by the Krebs cycle are easily utilized in the electron transport chain for the production of ATP. Now the details of how these ATPs are produced in the electron transport chain and how many ATPs are produced are here in this video about electron transport chain on our channel. In a nutshell, you can remember that about 2.5 ATPs are produced by one NADH and 1.5 ATPs by one molecule of FADH. Like we discussed in the summary, 3 NADH are produced through one turn of Krebs cycle and 1 NADH. So 3 NADH and 1 FADH produce 9 ATPs in total in the electron transport chain. Plus we also have 1 ATP that is directly produced in the Krebs cycle. Actually GTP is produced in the Krebs cycle here, and this GTP is further utilized to convert an ADP to ATP directly. Thus, one acetyl-CoA unit generates approximately 10 molecules of ATP through one turn of Krebs cycle. Since previously in glycolysis, one glucose was split into 2 pyruvate, which were further converted into 2 acetyl-CoA molecules, which enter the Krebs cycle. So 20 ATPs are produced per molecule of glucose in the Krebs cycle. I hope you liked the video.